individuals in animal cells, individuals are diploid. All of the life cycle is diploid. And it becomes diploid by the process of fertilization or conception when we have sperm and egg coming together, which are each haploid. So you have N plus N combining their nuclei to form this 2N single-celled structure, which we call what is formed when sperm and egg come together. N plus N equals a 2N cell zygote. We call it a zygote. So that's the way we remain for the rest of our lives, right? That zygote undergoes mitosis to grow and specialize and become all the different parts of the body. So every cell in your body is 2N, except for in the testes and ovaries, which are sperm and egg, which are N. In plants, it's not the case. In plants, there's two types of plants in the life cycle of a plant, two different forms, which we call the gametophyte and sporophyte. So phyte means plant. So when you hear phyto, think plant. That's the Greek or Latin term for plant. So we call that alternation of generations. So a generation is a life cycle, right? So when life cycles alternate between a haploid individual and a diploid individual, that's why we call that alternation of generations. And I have a picture in Blackboard I'll show you what that looks like. So the gametophyte might be larger or smaller than the sporophyte, so we don't look at size, we just have to look at chromosome number. So for example, if we look at a moss growing on a rock, we will find the green leafy part of a moss that you look at. That's the gametophyte generation. And mosses exist mostly as gametophytes, but every now and then, if you look closely at a moss, and do that this summer to challenge your biology skills and see if you remember this, you're going to see tiny little structures coming off of that moss that are the sporophyte. Have you ever noticed that? If you look closely, I'd like to be able to make this bigger, but let's see. I'll try this way. Oh, so much for that idea. I guess it's not going to work. But if you look closely, you know, I probably could even just search a moss and find a better picture. I don't want a diagram of a moss. I want a true moss. Let's take this out of here. No idea what that has to do with a moss, but there we go. Okay, so you've seen this on rocks before. If I look at any one of these pictures here, right, you've seen moss in the woods. But if you look really closely, you'll see that that moss has tiny, thin little stalks coming out of it, which is the other form that a moss can spend its part of its life cycle in, and it's called the sporophyte. So the sporophyte has the cells like we see in humans, the 2N cells that exist in the ovaries and the testes that undergo meiosis to make haploid cells. So the sporophyte has the cells used to make the gametophyte. But again, a majority is gametophyte. So here we can see those stalks on there. And in this picture, it's a little more realistic. You can see mostly gametophyte, the green short structures, followed by the sporophyte. So we only see that in plants. So animals do not have gametophytes. We exist as diploid individuals, and the process we use to get these haploid gametes is called gametogenesis. So another name for meiosis that occurs in ovaries or testes is called gametogenesis. If we're talking about the testes, we can further specify our terminology and call it spermatogenesis. So we get sperm from that. And if we're talking about gametogenesis in ovaries, that's called oogenesis, and that's for making egg cells. And we said there was a slight difference in humans with eggs versus sperm, in that females We know that at the end of meiosis, we're supposed to get 4 haploid cells, right? But in females, we don't get 4 egg cells. We get 1 egg cell in those 3 little non-functional polar bodies, because women don't need as many eggs as men need sperm. So this is the process side by side. It's all meiosis. It's still the same process we study, the generic process of meiosis. It's still prophase 1, metaphase 1, anaphase 1, telophase 1. Divide the 2 cells via cytokinesis, and then we have prophase, metaphase, anaphase, telophase 2, right? So all that still happens, no different, except for the end result is 1 egg, 3 little polar bodies. So that's what we see here. This first polar body is the first division. So instead of 2 separate cells, we get a nice egg cell and then a little polar body that didn't completely divide into a new cell. And then each of these is going to divide. So this divides into 2 cells, and this little polar body divides into 2 cells. So at the end, again, we have 3 polar bodies and 1 functional oocyte. But remember, we're stuck in metaphase 2 until females are stuck in metaphase 2 of meiosis in those egg cells until fertilization happens. So if you look and see here, you know, the green is meiosis 1, the yellow is meiosis 2, and then fertilization is what's needed to get that final structure called an ovum. So the ovum has already been fertilized and will become a zygote by mitosis. So fertilization, the sperm entering its nucleus and fusing with the egg nucleus, is required to complete meiosis 2. And that's when the final separation of chromosomes in that egg occurs. So if there's an error at that time, and it certainly can, we talk about with older eggs, there's more likely for the spindle to break and to drop off chromosomes or leave chromosomes behind. That's where we see a lot of genetic problems that people have with aging. So just be able to remember again, spermatogenesis, oogenesis are 2 examples of gametogenesis, which is an example of meiosis. They're all the same thing, just different terms, being a little more specific and telling us when and where this meiosis is occurring. So the way we say, geez, it's quite a party going on next door. Oa. Some people say ooh, and that's not it. When we see 2 O's, we want to say ooh, right? That's not how you say this. You say oa, oogenesis. Okay, so what happens when there's the wrong number of chromosomes because anaphase 2 of meiosis did not go well because of a broken spindle or some anomaly? Who knows? It's just a philosophy or just an idea of why chromosomal problems happen in older women. The thought is a broken spindle. So euploidy, U means good. So when you see the prefix EU, that means good. So there's something called eustress, which is good stress, which is the stress you feel when you're starting a championship game in basketball and representing your college. You get a little stress from that, but it also revs you up and gets you ready to play some good basketball, right? If you went out there lazy and lethargic, like you were going on maybe a required seminar for class, you know, you may not play very good basketball if you come in tired and lethargic, right? So U means good. So euploidy, we have the correct number of chromosomes if everything went well. Aneuploidy means there is some change in chromosome number. And what do we know if there's a change in chromosome number? It changes the organism significantly. So in some cases, it's just an extra chromosome, maybe in a cell that we have enough information to create the individual, but that extra chromosome or that missing chromosome causes physical, manifestations of that. So the phenotype changes. So if it's just one chromosome that's involved, we call it a monosomy, some referring to chromosome, and trisomy means we have 3 chromosomes. So Down syndrome is a good example of a trisomy. When a person has three chromosomes instead of 2, we're only supposed to have 2 sets. that means there's a problem. So non-disjunction just refers to the inability of chromosomes to separate appropriately, and then you have the wrong number of chromosomes. So here we look and we see this is normal meiosis occurring. So remember the tetrads form, and then at the end of meiosis 1, the tetrads separate, so you have a chromosome in each cell. And then in meiosis 2, remember the sister chromatids separate. Well, here, something happened in this cell, and when it split, maybe the spindle broke and it left a chromosome behind. So there's 2 instead of 1, and this one doesn't have any. So what happens then, so here's the sperm. What happens if this sperm, carrying 3 chromosomes, fertilizes the egg and infuses that into that zygote? Now that zygote has an extra chromosome that it shouldn't have. Or what happens here if that sperm enters the egg and inserts 1 chromosome? Now there's not enough information. And these are often common issues related to miscarriage. So when couples are trying to conceive and they find that, oh, we're just not getting pregnant, we've been trying every month, timing intercourse appropriately, it's just not happening, and there she's getting her period right on time. Actually, that female might be pregnant more often than she realizes, but because there's problems in sperm or egg, that there's not enough to develop and cause a pregnancy test to go positive. Because that early conception happens, the cells divide, but when there's not enough genetic information, there's not the recipe for making a human because of a lack of chromosomes or no chromosomes, then that can't develop enough to cause a pregnancy test to go positive, and a person gets their period right on time when actually they were pregnant multiple times in a year but just didn't know it. So that's why it's important. They tell young couples, I think it's under the age of 35, if they cannot conceive after a year of trying, they should see a reproductive endocrinologist. And an older couple over the age of 35, if they can't conceive after 6 months, they should come in and see somebody for testing, because older couples have less time. So they don't want older couples to wait if a full year if they might need some assistance. And we talked about stem cells, right? So what do couples do? They get genetic testing done, and they might find that some of their sperm or her eggs have chromosomal problems. They can do selection they can find a good egg and good sperm, whoever has the issue, where they can genetically pre-select the appropriate chromosomes, cells with appropriate chromosomes, and put those together outside the body. And what do we call that again? Do you remember? It's outside the body, so the prefix in vitro means outside the body. In vivo means inside the body. So in vitro fertilization is what they can do for some people with genetic issues. Sometimes it's in all their cells, and in that case, that you can't fix, and that's what makes people, permanently infertile, and then they have to use other options like donor eggs, donor sperm, adoption, or whatever. So, looking at trisomy, Down syndrome is a result of 3 chromosome number 21s. So that would be an example of non-disjunction like this, that would result in 3 chromosomes instead of, there's an extra, let's say if this was chromosome number 21, there'd be an extra one in there because of improper separation in anaphase 2. But because, I think I talked about this before, because chromosome number 21 is so tiny, it doesn't cause enough changes in the recipe for making a human that this person still survives. And we do see individuals with Down syndrome that live very productive lives. And some of the characteristics of Down syndrome is, developmental delays. So they have a childlike view of the world. In a lot of cases, that's desirable, right? Because they think everybody loves them, and they go through life with a very optimistic, positive attitude, and some are very, very happy people. So that can be a good thing. And there are some people that adopt, and they want a child with Down syndrome because the child, typically lives with them, through adulthood because of their, developmental delays. But some can even have normal intelligence, and and go on and, you know, live productive lives and hold jobs and all that. So, but there's certain characteristics. One of them, what they look for, like when I had my last child, I was 41, and the minute she was born, I saw them, you know, I was looking over, and I saw them look inside her palms right away, because they were looking for a sign of Down syndrome. Because when you look at a brand new baby, you know, the Down syndrome face is, you know, a flatter nose, puffy eyes. Well, a lot of babies, normal babies, are born with a flatter nose and puffy eyes just because of coming down the birth canal and the stress of delivery. So looking at the palms is an easier way to look for Down syndrome. So they have a single palm crease, which is characteristic of the 3 chromosome number 21s. But now I've had students look and say, oh, I have that. It doesn't mean if you have it, it's guaranteed, but it means if you look at every person with Down syndrome, they have the single crease down their palm. If you look at the general population, some will have the single crease, some won't. Does that make sense? So it's more, if everything else is there, the palmar crease is definitely there in a chromosome number 21 individual. And I can show you what that looks like too. So that's what it looks like. You go straight across. Most everybody else has an M. If you look at your hand, I can show you what that looks like. No, maybe not. No, it's not working. But anyway, most of us haven't. Here's an example of Down syndrome palmar crease straight across. Here's another one, goes straight across. Trying to find, here's the M. If you look, it kind of goes up and you'll see it come down and then make another M. It's kind of at a slant. Can everybody see that? Yeah? Okay, so that's genetics that put that M there. So characteristics of Down syndrome: short in stature, like very short, like an individual will be like between, not any higher than 5 feet, somewhere between 4 and 5 feet tall. They have a special fold over the eyelid, flatter nose, not much of a nose bridge, very short fingers, a very large tongue compared to their mouth. So a small oral cavity and a large tongue. So think of what that would be like for talking, for eating, for breathing. So oftentimes if you look at people with Down syndrome, they stick their tongue out a lot, and it's only because their oral cavity is so small. So they have issues with feeding babies with Down syndrome because of that. I think I talked about before, digestive and heart problems are really common. Cancers are more common in people with Down syndrome. So we see them in the hospital a lot. And because some are born to older parents, oftentimes they don't have any family because they were put into a home as the parents were aging. As they became adults, they were put into a group home, so they don't have much family surviving around, and then they end up in care. And I have seen, unfortunately, some people take care of adults with Down syndrome that are more profoundly affected, more severe developmental delays, can't take care of themselves. They use that as a source of income. And I've seen people with Down syndrome being poorly taken care of because the people just wanted the free income in taking care of these adults and not giving them good care at all. And it just really frustrates me. But if you have a compassion for people like this, there is a need for what's called respite care, where the primary people that take care of an adult with Down syndrome, sometimes they need a break so they can go out to dinner or just, take care of their marriage, have some special time with just the couple. So then people will take them into their home for the weekend. So if you ever have interest in that, there's always a demand for that. Other chromosomal issues are Turner syndrome. Turner syndrome is when, again, you have the splitting of the chromosomes, but the X chromosomes, one of them is missing. So that would be like this one here. Instead of this one here, instead of having 2 X chromosomes, one is missing, so they only receive 1 X chromosome. So maybe the sperm was missing an X chromosome, and in that cell, and the female, the egg, had the X, so they only had one X. And no Y. So if there's an X with no Y, it's a female, but there's characteristics that are, part of that. It's called Turner syndrome. So those people are short. They have abnormal moles on their face for some reason. We don't know why that is, but they too will have heart problems. And some can be of normal intelligence, some can have slight delays. I know a girl in my daughter's class that had Turner syndrome. I didn't know she had Turner syndrome because we met them, when they were in like 3rd and 4th grade. She would just look like a little girl in the class, and she was always doting over my younger children, and I told her mother all the time, like, oh, she's gonna make such a great mom, she's so maternal. And then I later found out she had Turner syndrome, and people with Turner syndrome can't have children because they don't have enough genetic information to conceive. So, because all of their cells are missing that X chromosome. Klinefelter syndrome is where you get the Y to make the boy part of the DNA, right? But then there's 2 X's, and that extra X causes problems with development. So because of that extra X is there, you have a little bit of more female characteristics. So they're not fully male. So they'll have underdeveloped testes, a little bit of breast development, long arms and legs, large hands. If you think about, some of your basketball players have Klinefelter syndrome. And who else has it? Michael Phelps has something called Marfan syndrome, which is a chromosomal condition as well. You've heard of Michael Phelps, the swimmer, right? So sometimes some of these chromosomal issues can give people advantages in athletics. So other types of problems with chromosomes are due to mutations. So there's different types of mutations. There's deletions that occur, and when would these mutations occur? When did we say mutations happen in DNA? When is DNA being adjusted in the cell cycle? When are we making DNA in the cell cycle? What do we call the process of making DNA from existing DNA? Replication, DNA replication. When does DNA replication occur? You gotta say it louder, I can't hear. I hear mumbles. Interphase, yeah, interphase, which part of interphase? Specifically, do you remember the 3 phases of interphase? Yeah, the S phase, the synthesis phase of interphase, right? So when the cell cycle is complete, and it's going to be ready to divide again, we undergo the S phase of interphase, we make more DNA. Well, sometimes DNA polymerase is faulty and makes an error in making a copy of DNA, and for some reason a piece of that DNA is missing. It's not replicated, so we call that a deletion. Or sometimes it's replicated twice, a piece of DNA, and now you've got a longer chromosome at the end, and that's called a duplication. A translocation is sometimes you have a segment of DNA moving from one chromosome to a non-homologous chromosome. So that can happen during what phase? When do we see swapping of DNA segments? Yes, prophase 1, right? Crossing over occurs. Well, there's other chromosomes in the environment, and sometimes that crossing over, that synapsis, happens between non-homologous chromosomes. So it's not chromosome 1 of dad and chromosome 1 of mom, it's chromosome 1 of dad and maybe chromosome 3 of mom, and now they swap and they have inappropriate chromosome segments on non-homologous chromosomes. So we call that a translocation. To translocate, just think of it like you're moving, right? You're moving from one place to another. Inversion is when there's a break in a chromosome and it's flipped. So now you don't have the right order of amino acids coming down that chain of DNA. It's been inverted, so it's flipped over. So these are all different types of mutations that can occur, and that's going to definitely affect the cell cycle and the outcome of the cell cycle.